Hi, Dr. Horvath. It's very great to see you again. So we are very thankful for you participating in our interview today.
Yeah,
So,
thank you for meeting me again, and I look forward to it.
okay, great.
Mhm. Mhm.
So, Dr. Horvath, it's a really honor to have you with us today. And thank you for taking time to speak with us. And your pioneering work on epigenetic aging, especially the Horvath Clock, has transformed the field of longevity research. Your contribution to understanding biological age have had an immense impact. So we are very excited to discuss your work today. To start, could you explain why the epigenetic approach is crucial in aging research? How does the methylation provide insight that other aging biomarkers may not?
Mm. Um, it is an interesting phenomenon that epigenetic changes to the DNA allow you to accurately measure time in all cells and tissues in humans, but also mammalian species. It's just a fact. And the question why that is possible probably relates to developmental processes. So epigenetic processes play a crucial role during development when the embryo develops. And in my view, aging is a continuation of development. And so therefore, perhaps these epigenetic changes allow you to measure time really from early development until late in life. I should say there are many different types of aging clocks. Other clocks use proteomics. They also work quite well. But proteomics clocks only work in one tissue at a time, for example, only in blood, but not in brain. And by contrast, methylation is unique in that it allows you to build aging clocks that work in multiple species at the same time. For example, we have methylation clocks that work for humans, but also for dogs, cats, pigs, really all mammalian species. That's very unique about methylation. Mhm.
Okay, great. So you are widely recognized for creating the first highly accurate DNA methylation-based aging clock. What inspired you to pursue this approach, and how did the idea originally come to you?
Yeah, well, by training, I'm a biostatistician. So I analyzed medical data for most of my career. And initially, I studied cancer and wanted to find ways to detect cancer early on. And therefore, I acquired expertise in building these biomarkers. And when I received tenure at UCLA and became a professor, I decided to go back to my dream as a teenager, which was to work on aging research. And I felt it was very important to have an accurate measure of aging because it's really a prerequisite for scientific study to be able to accurately measure aging. And I was very lucky because I had a colleague at UCLA who studied methylation, and so he had the data available. And I was able to use these data to prove to myself that it's actually very easy to develop a clock based on methylation. Mhm.
So you said that DNA methylation patterns will reflect the biological age. So could you elaborate on what this means in a molecular level? For example, how do methylation pattern of 30 years old differ from those of 60 years old?
Yeah, so when you look at methylation around the DNA molecule, you can visualize it almost like a landscape. There are mountains of high methylation and there are valleys of low methylation. And in a young person, you have these pronounced mountains and valleys. Whereas when we age, the landscape of methylation becomes more flat. So where you have high methylation, there's an erosion of methylation as we age. So methylation decreases. And conversely, regions where we want to have low methylation, they often gain methylation with aging. So, and these age-related changes take place in millions of locations on the DNA molecule, both age-related gain of methylation and age-related loss of methylation. And these epigenetic clocks that we have developed typically look at several hundred locations, half of them that gain methylation, half that lose methylation. And they look at these hundreds of locations and form an average. And this average allows you then to arrive at an age estimate for a person, or it allows you to estimate their mortality risk. Mm-hmm.
Okay. I mean, so you mentioned about you're looking at several hundred methylation sites, and your 2013 paper that introduced really a paradigm shift in aging research. So I understand that while the telomere-based aging measurement had a correlation of about 0.5 to 0.6 with the biological age, and your clock actually achieved an astonishing 0.96. I found out that some of the reviewers were so surprised that they delayed the publication. Could you share your experience of that time?
Yes. So when I claimed that you can build a clock that has an accuracy of 96%, most people simply couldn't believe it, including myself, by the way. I need you to know that. I also couldn't believe it. And many people assume that an error must have been made in the data analysis, because sometimes people inadvertently bias the analysis, So I remember one reviewer rejected the paper and said it cannot be true, because if it was true, it would be a revolution in biomedical research. But this reviewer simply couldn't believe it, And I need to tell you, after I published the paper, for the first 3 weeks, I was also very, very nervous about the claim of accuracy. And I was then relieved when other people took my clocks and applied it to their data and validated the claim, So, but yeah, it was difficult to publish the paper. But I think many scientists go through that, So papers get rejected multiple times. It's just very painful to publish. Mm-hmm.
What makes your clock so uniquely precise? Is there a specific reason why it could achieve such a high accuracy compared to other aging models?
Yes, there were several reasons. So the first reason is biological. So these epigenetic clocks relate to fundamental developmental processes that are very precise. So there's a strong signal. And what do I mean by that? Remember how I said there are certain regions on the DNA where you have very low methylation and others where you have very high methylation. And this signal is very strong. So the second reason why epigenetic clocks work so well is that it's very easy to measure methylation. What do I mean by that? Imagine you draw blood and you forget to put the blood tube in a refrigerator. You leave it outside and the blood coagulates, Now, this heat treatment would destroy the signal in messenger RNA or many other readouts. But interestingly, methylation is quite robust. I mention it because that's exactly what happened to me. I transported blood tubes in a car, and it was a hot day, and I could still measure methylation. So DNA methylation is very stable, you know. And the third reason why methylation clocks are so accurate is that many data are available. So when you're a data analyst, you have access to large data sets. And I was very fortunate in that regard because back in 2012, I downloaded publicly available data. Anybody could have done what I had done because the data were on the internet, you know, and I had access to many thousands of samples, you know. So yeah, these are the main reasons, I think, why methylation clocks are so accurate.
Okay. So since your initial discovery, aging clock have evolved significantly. Could you highlight some of the key advancements in the field over the past decade?
Yes. So our initial clocks measured calendar age, simply how old is the person. And then the next big development was to estimate time to death. So how much time someone has left on planet Earth. And this was done by looking at so-called retrospective cohort studies, where people had collected blood samples in the 1990s, and then they followed these people and knew whether they still were alive or dead, what they had died from. So they could look back in time. And with these data, we and others were able to predict mortality risk, you know. The next big development were clocks that apply to many different species. So the first clocks were developed for humans, but then people developed clocks for dogs, for mice, for cats. And last year, we developed so-called pan-mammalian clocks. We have a clock that measures age in all mammalian species, you know. More recently, people developed clocks for single cells. That's also very exciting. So we can measure the age of a single cell in your body.
So you and your colleagues conducted a fascinating study where the participant received some human growth hormones and metformin for about a year, and leading to an average reduction of 2.5 years in biological age. Could you walk us through this study and share your thoughts on this groundbreaking research?
Yes. This was a very small study. I need to emphasize a preliminary study where my collaborator, Greg Fahey, analyzed 9 men, and he administered a cocktail of rejuvenating interventions. So a growth hormone, metformin, vitamin D, zinc, and other compounds in order to rejuvenate the immune system, the so-called thymus gland. And in this small study, we observed that epigenetic age was reduced by roughly 2.5 years after 9 months of this treatment. So that was encouraging. But I want to tell you and the audience that growth hormone treatments are quite risky. And these kinds of treatments should only be taken under the supervision of a doctor. But my collaborator, Greg Fahey, carried out a second study with more people. I want to say over 20 people. And he recently mentioned in a scientific conference that he was able to validate the original claim of age reduction. So these results are encouraging, but I need to mention they are preliminary and they still require more testing.
Yeah, I mean, it is preliminary, but it is very promising that, now we have something that we can really, reversing the aging by some science and interventions. Okay,
Yes.
so for the general public, what are the practical implications of knowing their biological age through the aging clock studies? How can this information really help individuals to make better health and lifestyle decisions?
I think the main purpose is to inspire people to follow a healthy lifestyle. So, for example, people know that they should avoid sugar, they should avoid obesity, they should stop smoking, they should walk or exercise a bit. People know all of that, but it actually takes a lot of discipline to follow a healthy lifestyle. And maybe such measurements can inspire people to follow a healthy lifestyle. I want to tell you, I do not need such measurements, because I know that I have to eat vegetables and be healthy. I don't need to measure my age, so to speak, but I've heard from others that it inspires them when they get a methylation age estimate, But so that's the here and now. My hope is, of course, that ultimately methylation clocks will inform the decision maker such as a doctor. But I need to mention that we are not there yet, because we do not yet have medications that reverse methylation age. But my hope is that researchers will find medications that reverse methylation age. And if that is the case, then it would make sense that a doctor prescribes a methylation age test in the clinic.
I mean, it's very promising that soon or later, we may have medications or interventions that can really reverse the agings. So where do you see the field of methylation aging clock heading in the next 5 to 10 years? Are there any emerging technologies, applications that really excite you the most?
Yeah, the field of methylation clocks is very vibrant. I want to say dozens of research groups are working in that field, and they're developing better clocks and cheaper clocks. So when I measure methylation age, it's actually quite expensive for me, a couple of $100, and that's too expensive. So some people develop cheaper methylation clocks. The second line of research focuses on what one could call causal epigenetic clocks, and causal for aging. Because, as I mentioned, millions of locations on the DNA molecule change with age, but not all of these changes lead to organ dysfunction. Not all of these changes are causally related to aging. And this gives rise to an idea. Focus on the locations on the DNA that truly cause aging. And so many people work on these causal aging clocks. And the third line of research is to adapt methylation clocks for pets and animals, because we want that our dogs live longer and cats live longer. So there's also a research in that arena.
So your pan mammalian should work with all the companion animals that people really care about. Okay.
Yes.
So my last question is, in recent years, there has been an increasing interest in aging and longevity research in Asia, particularly in South Korea. So in your view, why is Korea is important player in this field, and what unique strengths does Korea have in aging science and longevity research?
Yes, I think Korea can play a really crucial role in advancing aging research and longevity research for many different reasons. First and foremost, Korea has an aging population and needs to help to find a solution. And second, Korea is a highly developed society with access to medical records, and also has the resources to fund research to identify particular opportunities for rejuvenating people. So, for example, people need to work on different organs, for example, the brain, dementia, or the lung, or the heart. And so there's so much work to do. So it's important to characterize these diseases very carefully, then collect blood from these individuals so that we can understand which biomarkers and which clocks can predict health outcomes. Korea can help with both human studies, but also animal studies. And finally, I want to highlight the tremendous expertise by Koreans in technological products. So, for example, wearables. We can measure motion and develop new biomarkers based on that. Yeah. Mm-hmm.
Can you share with us that what kind of anti-aging or reverse aging you personally practice? You mentioned about you are eating some healthy food, but can you share with us a little more detail what you are doing to reversing the age or the anti-aging?
Sure. Yes, I try to stay young. It is an obsession of mine. So I think about staying young 24 hours a day. For me, it all starts out in the morning. So I start out by jogging on a treadmill for about 10 minutes. Then I lift weights for about 10 minutes. Next, I eat a very healthy breakfast, which is 2 eggs and a lot of broccoli. So I have a lot of broccoli and other vegetables like Brussels sprouts in my freezer, and I take them out, put them in the microwave oven, and have a lot of green leafy vegetables with olive oil, 2 eggs. That's my breakfast. Then for the rest of the day, I try to avoid or reduce carbohydrates. I do eat carbohydrates, but I just try to avoid sugar spikes. Then towards the evening, I try to calm myself down in order to improve my sleep. I struggle with sleeplessness, so I'm doing my best to calm down. Usually, I walk after dinner, not for long, maybe 25 minutes or so, just to increase my step count. So I count steps and I try to walk about 8,000 steps a day, apart from the jogging in the morning. Then I take various supplements, creatine. Then I take vitamin D. I take omega-3 fish oil. I take magnesium. And yes, I suffer from high cholesterol levels. Therefore, I take statin and another compound called ezetimibe. So these are 2 drugs that lower cholesterol levels. And that's about it,
Thank you for sharing with your secret recipe. It is very helpful. And foremost, I think, eat healthy and exercise and sleep well. I think that's the most important lessons that we all have to learn.
I agree. I agree.
So do you have any other things that you want to mention or comments?
We live in very exciting times for many reasons. Above all, the science has progressed rapidly. We understand quite a lot about why we age and what can be done against it. At the same time, there have been technological breakthroughs. And I'm very hopeful that significant discoveries will be made by the biomedical research community. And so I want to tell everyone, stay healthy. Many exciting medications will be developed by the community. So,
Great. Dr. Horvath, thank you for sharing your invaluable insight with us today. Your work has not only reshaped how we understand aging, but has also opened the door to exciting new possibilities for health and longevity. Once again, we deeply appreciate your time and expertise. We look forward to featuring your groundbreaking work in our documentary and sharing it with our audience. Thank you so much.
Yeah, thank you so much for your kind words, and I wish that you stay young and healthy for many years. Thank you. Thank you again. That was fun. All the best to you.